So this is cellular respiration, and let us start with the molecule of glucose. Now, before we enter the mitochondrion, the glucose undergoes a 10-step process, shown here, uh, that breaks it down. It's called glycolysis. So at the end, you get two pyruvates, a 3-carbon molecule, two ATPs, and two NADHs. Now, these pyruvate molecules make it into the mitochondrion, and specifically through the outer membrane and through the inner membrane into the matrix. Now, from this point onwards, we're gonna track one of the pyruvate molecules. So, inside the matrix, the pyruvate undergoes oxidation and is attached to a CoA molecule to form acetyl-CoA. The CoA molecule is just short for coenzyme A. So this 2-carbon molecule floats around the matrix, and at the same time, 1 NADH is produced, and 3 carbon becomes 2 carbon. So 1 carbon is lost as carbon dioxide. Oxygen is involved here. Now, then we enter the citric acid cycle, or called the Krebs cycle. Really, there's another name for it. It's called, if I'm not mistaken, never mind. Just forget about it. It's called the citric acid cycle, or the Krebs cycle. Now, the citric acid cycle starts with this molecule called oxaloacetate, which is a 4-carbon molecule. So this acetyl-CoA combines together with oxaloacetate via this enzyme called citrate synthase to produce citrate, which is also citric acid. Now, this citrate, via the works of other enzymes like cotinase and isocitrate dehydrogenase, produces one NADH and another CO2, which means it turns into a 5-carbon molecule called alpha ketoglutarate. Now, this alpha ketoglutarate bonds with the coenzyme A again, and with the help of alpha ketoglutarate dehydrogenase, forms succinyl-CoA, and in this process it liberates an NADH and a CO2 again, which means it's getting reduced into a 4-carbon molecule, and the succinyl-CoA turns into succinate, so meaning the CoA is recycled again. And then it liberates a GTP molecule. Now, the GTP, like ATP, it's a nucleoside triphosphate. It's an energy carrier. And it has similar functions and similar roles as ATP. It carries energy, but GTP is primarily used in chemical signaling pathways. Now, here we have succinate, and then succinate, it goes to this complex called succinic dehydrogenase, or known as electron transport chain 2, and it's converted into what's the name again? Into fumarate, via the help of fumarase again. They both are in the same place. And then this liberates to FADH2. No, a single FADH2 molecule, which also carries hydrogen ions. Now, this fumarate molecule, via the malate dehydrogenase enzyme, gets turned back into oxaloacetate, which is our starting molecule. And in this conversion, an NADH is liberated. So, to recap, 10 NADHs are produced per cycle. 2 FADH2s are produced in a single with a single glucose, and then 2 ATPs, 2 GTPs, and 6 carbon dioxides. It makes sense because glucose gets completely oxidized into carbon dioxide. Well, it ain't strange. So then the next thing that happens is this electron transport chain. Thus, electron transport complex 1, 2, 3, and then 4, along with 2 electron carriers, ubiquinone, and then cytochrome C. So previously we have mentioned that succinyl-CoA turns into succinate, and then the succinate turns into fumarase via the help of the electron transport complex 2. So let's start from there. What precisely happens is that after the conversion, the FADH2 enters the ETC2, which donates 2 hydrides. That is hydrogens with their, not exactly hydrides, but 2 hydrogens. It's less confusing this way. 2 ubiquinone. So this ubiquinone, in its form QH2, transports the electrons to ETC number 3, which is this one. Now, at the same time, the 10 NADHs, they get converted into NAD pluses, and also 10 H pluses. So this provides energy to pump hydrogen ions from the matrix to the inner membrane space. Now, the electrons are shuttled via this ubiquinone again, and then the ubiquinone accepts 2 H pluses from the matrix to form this QH2 again, and then it transports the electron to ETC number 3. And with these electrons, ETC number 3 pumps H plus ions into the inner membrane space, which then the electrons gets transferred to cytochrome C. Cytochrome C transports these electrons to ETC number 4, which is the place where H plus ions and oxygen gets reduced into water, hence balancing the whole equation. So it is estimated that for every NADH molecule